All right, today we're going to quickly go over the process of transcription and translation and why that is one of the important reasons behind, or the main ideas or the central dogma behind molecular genetics. Let's start with some of the things we already know. We know that we've got DNA, and DNA is a double-stranded, we call it a double helix. The process we want to talk about today is how that DNA is going to end up coding for the structure or all the proteins that we have in our body. Now, we want to take that DNA strand and somehow make that strand a single-stranded RNA molecule. So we're going to talk about how that process is going to take place, and then from there, how that that RNA structure can then convert it into a protein. Now, the process that's going to be taking place here at that site is known as transcription. Okay? The process that is going to be taking place at this site is known as translation. Transcription and translation are the two different things that we're going to be covering here. How do I convert DNA into RNA, transcription, and RNA into protein translation? One of the last things we can take a look here is at where are these certain things taking place. If I do a quick little illustration here, these things in this pink box are actually going to be taking place, let me shrink that down, in the nucleus, and then the conversion of RNA into a protein. Let's select a different color here. Like this rainbowy looking color. This is gonna, looks like it's the same color. That section there is going to be taking place in the cytoplasm. Okay, so let's move our way into a new slide. So what is it actually taking place? Now what we have finished, we've talked about in DNA replication, they had to use 3 enzymes. If you remember, those 3 enzymes were helicase, DNA polymerase, and the third one was ligase. We're actually going to mix all that together, and we're only going to do one enzyme, and that enzyme is going to be RNA polymerase. It's going to do the job that all 3 of these things kind of did. What I'm going to look to do is I'm going to take this DNA strand that I've got, and I don't actually want the whole thing copied. If you think back to how we selected this thing in DNA replication, we copied the entire thing. We split them apart. In fact, let's take a look at this a little bit different. We had one strand here and one strand here. Those 2 different strands. What I ended up doing was having a strand there, strand here, original strand, and there. I ended up with 2 identical strands here in that semi form. But all I want to do is not copy the entire structure. What I actually want to get done is I only want to take a little section of DNA. So if I go back to my DNA strand, if I've got this DNA strand here, I only want a little section of it. So maybe I want this piece right there. So if I zoom in on that piece and I explore this down here, I'm just going to kind of zoom in on that box. What I want to do is actually take one little section, or the piece that I want to zoom in on is just one of those little pieces in there. So if that piece, let's go back to our white line, was listed here, and maybe our letters are shown here. Let's just do some quick. A, T, C, C, G, A, T. Say those are our letters of the DNA that we want to do. Now, RNA polymerase, the job that we had talked about up here, is going to open up that DNA strand. What it's going to do is it's going to read those letters, and it's going to take those letters and figure out the copies of those letters and end up giving you, kind of open this back up. But we've seen this before in terms of DNA replication, right? We're going to just copy the certain letters that we actually want. Right, and now if our white end was more of our 3' end, here's a 5', more 5, it's more than 3. We're going to develop a brand new strand, and it's going to go in that direction. Now, that little strand, that is the section of DNA that we want that is considered our gene. So how do I actually code for it? RNA polymerase comes in and puts down the corresponding letters. So if those green letters here were A, T, C, C, G, A, T, T, A, C, C, G, I want to read those in terms of RNA. Now remember, some of those rules about RNA does not have any T's. So if I read those correctly, our new strand of RNA is going to read, by reading those letters, A, C is an A. In DNA, would put down a T, but it's actually going to put down a U for us. C is the second letter, which is T. What corresponds with T would be an A. Third letter there is a C. What corresponds to that is a G. Fourth letter there is another C, so that would be a G it corresponds to. Fifth letter is a G, so that would be a C. Here we've got an A, so that would be correspond with a U. T with an A, another T with an A with a U, two C's and a G, so you got G, G, C. That is now my code or my message that I am going to take out. That is just a small section of DNA that is going to code for our protein, and we consider that our gene. Okay, now if we move on a little bit further, if I have that one section of message, so there's our message. In particular, we name this mRNA, our messenger RNA. Now, what the job of this messenger RNA, when it was transcribed back here, this whole process, remember, is taking place. If I'm going to zoom in on a cell, let's draw something here. Here would be in the nucleus, and I'm in the large actual cell. So remember that transcription is actually taking place there inside the nucleus. Okay. Now, this important message, let's give us a different color here. This important message that's shown right there is going to find its way out, that being our mRNA, is going to find its way out, and that's showing us here. Now, its job is to find it's going to find a ribosome free-floating around in the cytoplasm. Now, it could be free-floating, I guess, or it could be actually on an endoplasmic reticulum, making it the rough ER. But what we're going to do is we're going to take this message, and we're going to read it 3 letters at a time. Okay? If we have our particular letters on our mRNA sequence, we're going to have a whole bunch of A's, we might have U's, G's, all sorts of different letters here that's going to correspond to it. Just any randomized letter that I'm going to put in right now. What it's going to do, it's going to find its way through and land inside of what's considered a ribosome. And a ribosome's really got 3 initiation spots. It's going to have an arrival site, a placement site, and an exit site. All right. What's going to correspond with this guy is that you're going to see free floating around the cytoplasm another little structure, which is a tRNA that's going to have also 3 little spots on it, and attached to it is going to be a particular amino acid. So what's going to happen is if I take this mRNA sequence, now that amino acid is going to be important in the assembly of our particular proteins. So now what's going to happen is we're going to take this structure here, and it's actually going to read and going to land on the A site, or the A for arrival site. As it works its way through there, our RNA reads 3 letters at a time. 3-letter sequences, so if you take those first 3-letter sequences, a 3-letter sequence is considered a codon. Okay? Now, if you look down at this tRNA structure, it looks like a T, that's our tRNA, it's going to transfer in that a particular amino acid that's shown there. Now, these 3-letter sequence that's on the bottom is going to be the anti-codon. So if I'm looking at this structure up here, this AUG, that AUG that's illustrated there, I want an opposite of what an AUG would be. So that would be an opposite of AUG. So if I'm looking at this and I have an AUG, I would want an opposite of that. So I'd want a UAC on my anticodon. So what I'm going to do is I'm going to take this structure and just kind of move it off to the side a little bit. I want to take a particular mRNA sequence, floating on in. We're going to say read 3 letters at a time. 3, so on and so forth. Here's the bottom part of a ribosome, and here's our top part. We got an A site for arrival, P for placement, or E for exit. If our first 3 letters, and you're going to learn why I'm always starting with an AUG here later on, AUG is always going to be our start codon, is going to come in. As it reads those first few letters, that codon, it comes through, and a tRNA with an anticodon is going to stick like a puzzle piece to those guys. And what it's going to bring is, it's going to bring its particular amino acid sequence. And that amino acid that we know is called methionine. Now you're thinking to yourself, well, how do you know that? In our next slide, I'm going to show you an illustration here quickly. Pop that up. So AUG, how do I know it? So here, AUG, there's our actual code. Now let's utilize this tool on the bottom. Find the biggest A. It's going to be located in the center. And so I want to stay in this quadrant. There's my A. The second letter is a U. All right, there's the U. And the third quadrant is the G. So there's our methionine, our term methionine that we need to refer to. Now, I can give you a different one. Let's try CGA. See what that codes for. Find the C, the largest of the C's. Okay, it's there. Find the next letter is a G. That's there. And A. That's in there. So this one would actually code for arginine. Okay, the amino acid sequence there. So those are what we're going to be looking at in terms of our sequencing. All right, a few other things that you want to take a peek at here in the sequence are the note that you've got multiple spots, that you've got more than one ability in that third letter to have the same amino acid. If I had only one of these letters, if this is G and C, coded for one amino acid, we'd only have a total of 4 amino acids. Now, these are the letters that we have in terms of our nitrogen bases. If I only use 2 different combinations, A could be with U, A could be with G, A could be with C, A could be with A, there would be 1 set. We could have up to 16 possible different combinations. But remember, in terms of how many amino acids we have 20 different ones. So if I use it up to 3 letters in length, we're going to have a whole bunch of options. That's why you see some of these have multiple different endings, even though it still codes for the same amino acid. Okay. Now the last thing I want to take a look at this wheel is that we've got 3 different amino acids that are actually going to be used as considered a stop. Now the stop codons are going to be codons that actually do not have an amino acid sequence whatsoever. So what's going to happen is if we're looking at our ribosome that is assembling these guys, it's going to see the 3 different ones. Those 3 different ones are UAA, UGA, and UAG. You find these guys over here. Okay, those are our stop codons. When we look at a structure, let's go back to our. Let's actually make a new real quick. Shrink that up. We had our protein, our ribosome here, being developed. And as the mRNA sequence is riding on through here, reading through our letters, all of a sudden here was a UAG, one of those stop codons. There is no tRNA for it. So what happens is that the ribosome breaks apart. It disassembles. And what's left over is all the amino acid sequences that they had beforehand. All right, and that's where we're going to stop for today. We're going to come on back and we're going to take a look at how those amino acid sequences are, again, going to be used in that development of the protein through those peptide bonds.